Hello, welcome to section 2 of learning and memory. In this section, we're going to speak about how the brain acquires new learning, exactly from sensory registering to encoding, from the time that the information comes in contact with our senses, with our sensory channels, to the time that the information is taken in, is processed and broken down and processed and taken in. So information processing occurs along many stages, many steps. You can call them phases because the word phase implies change. So first, when the information comes in contact with your sensory mechanism, you see something. When you see it, it has now, your eyes have contacted certain information. Maybe you saw someone. Maybe you saw a star or a cloud or a tree, whatever it is. That is sensory information that has come in contact with your sensory channels. Sound coming in, and then once the sound wave comes in, and then you hear the sound, then you have registered the sound. So registration doesn't mean that the information is processed. For example, you might be sitting down somewhere and some noise is going on. Like say a bus parked next to you, not far, and it's making a lot of noise. Initially, you are going to hear that noise, but in a few seconds, 10, 20 seconds, you are not going to be aware at all of that. So even though your sensory mechanisms are receiving and registering the information that's coming in into your ears, that information is not encoded. It's not going to your higher brain levels. The brain is not going to care about a signal that continues to go the same way. So changing signals, changing your tone of voice, the way you are doing things, moving around, that is the way strategies you can take and develop based on that idea that if a sensory mechanism, information is not changing, then the brain will not exert effort in processing it. So then, encoding and transfer and storage, retrieval and consolidation. The centers we are speaking about are in the core of the brain and also in the temporal cortex above the ears. In that temporal cortex, you have your memory center deep in the temporal lobe, and that is the hippocampus. We mentioned it before. And then near it, you have the emotional processing system, which is the amygdala. And then across, you are going to have areas like, for example, the basal ganglia. You are going to have the thalamus. Yeah, they are not showing here. But a whole system that exists in this here, that is called, traditionally, called the limbic system. That includes memory, learning, motivation, and emotions. And the frontal part of the brain supervises everything, and it kind of directs what is going on in these areas. Even as your hippocampus is trying to facilitate the storage of some information, it has to work back and forth with the prefrontal cortex to determine what needs to be stored, what should be discarded, and so on. So learning, things that we learn every single day, things that you do in your classroom or in any kind of a situation, that new experiences, new learning modifies our brain. The physical construction, the physical structure of the brain changes as a result of the learning that we go through, that we acquire. So we'll go over a few examples of these changes. First, the physical structure of the brain, how it connects. And that's the most important thing, is connectivity, not how many cells you have. It is how the networks, how neurons connect to make circuits and networks to make skills possible to learn. So these the structures of the networks will be modified, will be changed as a result of new learning. And that change is going to involve electrical, which firing, biochemical, changing the chemistry and the biology, the nature of the cell itself, and genetic mechanisms. When we say genetic mechanisms here, I want to say that, let's go back to this image of the hippocampus and the areas around it here. The hippocampus and the areas around it, as we mentioned way back at the beginning of the course, that is one of two areas, and this area is the primary one that is known to have neurogenesis. It gives birth to new neurons. And these neurons are important for us to learn new skills, and they are embedded, they are used to build networks and circuits and to connect new information to old information, and also to replace the neurons that die because of normal processes. So in this, how these neurons are produced, the new ones, how are they produced? The body of say when a nerve cell is going to divide, or any kind of cell is going to divide, I mean not a nerve cell, but the cell that gives that is the progenitor cell that produces, out of which neurons are produced. It has DNA, and the DNA is copied, and then you produce kind of a multiple duplicate the DNA, and you create cell division and all of this stuff, so you can produce the neuron. So you need DNA translation, in other words. That is the genetic part that I'm talking about. There are many agents to contribute to the development of pathways and to the development of neurons and the maintenance of the health of the neurons and so on. One of these are called brain-derived neurotrophic factors, BDNF. If you Google that, you'll find numerous articles speaking about how these are essential for building brain structures, for maintaining the health of the brain, for enabling us basically to maintain producing new cells and learning new stuff as we go. So BDNF is critical for synaptogenesis, making synapses, connections between neurons, and synaptic plasticity, making it possible that, if this connection is not used or doesn't work, we can sever it and we can make a connection somewhere else. So that is synaptic plasticity, being the flexibility and the wiring of the brain. It contributes to presynaptic release of glutamate, and that's a major neurotransmitter. That's the most common neurotransmitter in the brain. It contributes to consolidation of new information through making some changes in the number and in the shape of the dendritic spines. And also in reorganizing the subsynaptic actin cytoskeleton, which means the cytoskeleton, let me just go here. These are the synaptic, the dendritic spines we discussed before. These little budding, pieces. That's where neurons connect with other neurons to make synaptic connections, and that increases the surface area. And one neuron can, for example, may be able to synapse with 50,000 other neurons because it has so many spots. So this is the synaptic complex. And what we mean is the area here, the receiving neuron has channels, receptor channels, and the neuron that's sending a signal sends neurotransmitters that are released in the cleft between the receiving neuron and the neuron that is sending. So these two components and the cleft in between, these make the synaptic complex. The modifications that occur, these channels where the neurotransmitter docks, and then it causes the channel to open or to close. And then once you have the information going through, once it has achieved its purpose, then the neurotransmitter takes off and either disintegrates or goes back to channels so that it can go back into the cell that sent it, and it goes again to be reused again. Or it can be degraded and recycled again or assembled in many cases. So what I am saying here is the number of receptor channels determines the efficiency of a signal. The number of vesicles and the amount of neurotransmitters that this neuron that is sending the signal has is going to determine the efficiency of the signal. Now, as we get the neuron to fire over and over and over and over, the more we get a neuron to fire, the more likelihood it will continue, it will fire again, and it will fire more efficiently. Why? Because it learns how to send these vesicles quickly, how to package the neurotransmitter in that quickly, how to have enough neurotransmitters. So the more frequently you get the signal refreshed and you hear something, for example, or see something or do something, the more neurotransmitters and the faster that they are processed so that they are released, and then the more receptor channels that you are going to have along the cell membrane of the receiving neuron, and that is going to make the signal very efficient, and that's what makes it automatic, the skill automatic and spontaneous and so on. So these are physical changes that if someone is not practicing a skill, or there has been a very long time when they kind of revisited the information or whatsoever, these numbers are going to decrease again, and this is not going to be active much, and it will only, as time goes by, it will eventually be degraded, it will be disconnected, and the thing is, all the information on it will be gone. So these are physical changes that happen on the synaptic level, on the cellular level. You have bigger changes in addition that happen on the level of the network itself, on the circuit itself. It will be modified. So acquisition of new information and memory formation is modified by the formation of new active synaptic connections. So you have now new synapses are made, and the more they are stimulated, the more that they are going to stay, and the more channels they will have, the more neurotransmitters they will have, and the faster neurotransmitters will go back and forth. Also, new experience induces firing. So new experience stimulates the firing of neurons, so that the only way they know how to connect well is by constantly being activated. Neurons that fire together wire together, because when they fire together, they have signals to identify each other and see that they have the same function, that they basically function, carry out the same job. So they start to connect and identify each other when they fire. Continuous firing leads to strengthening of synaptic connections among neurons that share the same function. So in other words, take it from your perspective. When you teach someone something, review it over, have the person practice. This is when we say practice makes perfect, because these changes do happen to the structure of, of the neurons and the circuits themselves. So the neurons then, through the firing process, they identify each other, and they know that they are specialized in the same function, and they connect and they form a network. Over time, that network will be fine-tuned, and it will be kind of more specialized and efficient. The more it fires, the better it connects, the more efficient it will be over time. So listen to this. Persistent synaptic activation through stimulation, like saying something to someone or teaching a child or speaking, communicating, anything that you engage in at all. Persistent synaptic activation means repeated experiences from the outside world lead to a number of pre and post synaptic molecular events. Pre means in the neuron that is sending the information, and post is the neuron that is receiving, the one that has the channels to receive to open for the chemicals that come in to carry the signal. So, and the one that is sending, that has the neurotransmitters and components to send the signal. So modification will happen here and will happen here. And these include release of inhibitory or excitatory neurotransmitters, whether neurotransmitters to make the gate, the channel close, or to make it open, inhibitory close and excitatory open. And increase receptor expression, which is these channels that can increase as a result of the repeated stimulation, persistent, on the postsynaptic neuron, resulting in the activation of distinct signaling pathways. So you are now, the more you practice a skill, whether reading a story, whether hearing songs, poems, playing a game, for acting in a play, whatever, the more you do it, the more these changes will fine-tune your system. That is going to result in the activation of distinct signaling pathways that temporarily and permanently alter the specific patterns of gene expression. So now that the machinery is working fast and efficiently, that is going to go back to the factory to say, okay, we're working quickly, you need to give me more raw materials, you need to give me whatever it takes to sustain this performance. And now genetic expression and the production of new products is going to be faster as well, to support the new changes that are taking place and to basically modify and reconstruct these networks to make them more efficient. Cellular changes involve modification. Number one, on the synaptic, modification in the synaptic strength. How strong will this synapse be? There are molecules that are called adhesive molecules that cause the two parts of the synapse to stay together. So that will be strengthened. Synaptic plasticity, the flexibility that when there's a change of a connection or making a reconnection, that is possible. That increases the possibility. And synaptic consolidation. So that applies to certain synapses that are known to, now they are connected well, they are continuously stimulated, they will be consolidated, they can hold on to the information for a long, long time. That is coupled with intrinsic, dendritic, or somatic plasticity. That means the body of the neuron itself, that is the soma, the body itself is going to be modified in a way as to support the new skill or function. And dendrites are going to be modified as well by having more dendritic spines, by having maybe taking away some from one area, putting them in another area, and so on. So to modify, that is going to be modified based on the nature of the stimulation that you are getting. And you can translate this into, and then based on the nature of the teaching that you are engaging your students in, based on the kind of skills that you are practicing, and so on. So you can see directly from outside, you are modifying the physical structure of the brain. So now we're going to speak about how the brain receives and how it encodes new information. So let's look at the first stage of learning, which is sensory registering and perception. So as I explained, once our senses, whatever senses, when we speak about senses, you think about the five that we all know, the famous ones. But there are the rest of them, which is they are all about 21 senses. So once our senses come in contact with information, then that is sensory registering. When the information reaches the brain, the cortex, then we kind of understand, we process that information, we become aware of its presence, and we begin working on interpreting it. That is perception. So when we say something is perceptual, it implies that it varies from person to person because our sensory mechanisms are not the same. The way your hearing is, depending on how your hearing level, then for example, how your background knowledge helps you interpret the information that is coming in, your environment, everything else, that makes our sensory channels and our perception will be different from person to person. So then the information, once it reaches the cortex, it will eventually go to the hippocampus. So as it goes to the hippocampus, you sustain information in short-term memory, in working memory, and at the same time you have mechanisms of attention and you have mechanisms of executive functions that will focus your attention on that information. And also this is your attention and focus are driven by your motivation, by the way that you feel toward your goal, what you want to achieve, what you want to do, what you want to say. So that is your goal. That is, it is based on your motivation. And that is going to help you direct your resources, your cognitive resources. For example, planning in order to achieve the goal, planning resources, planning your time, planning how to do and when to do, certain steps and so on. So all of this starts at the very beginning of the encoding process. Once you have attention in place, executive functions in place, working memory systems in place, now you have the information sustained. You are ready to take pieces of it, break it down, means analyze it, and then kind of figure out the relationships between different parts and components and so on, and reshuffle and synthesize, get new ideas out of that information and so on. So the hippocampus serves as the mechanism to kind of where information for the most part just goes in there and it will be sorted out. It will be broken down, will be analyzed. I tell, when I speak about this area, I liken it to a post office processing center. At some point, I went to a post office facility to receive my mail after I came back from a trip. and they have it like in a very big, wide area, in a big, like a large storehouse. And there's an assembly belt that goes very big and goes like in a rectangle, but extremely large. And you have people come and dump baskets of mail, and someone takes these letters and envelopes and flips them so that they would show the barcode. And the belt, as it continues, it is scanned, the barcode is scanned, and then that letter is going to go to a particular area, as the belt moves, it's going to be directed to a certain area. It's going to fall into a basket, and then that is going to go to a particular town. So, but initially, when all this mail comes in, it is not distinguished, and then as it goes through, it becomes processed. So that is a mail processing center. The same way like what happens with hippocampus. All information, converges on it, and as it's coming in, and then it is analyzed and sorted out and connected, and connected with the storage sites above. So perceptual encoding is perceiving sensory stimuli, transforming them into cognitive representations in the medial temporal cortex. Medial temporal cortex. So temporal cortex has 3 lobes, and in the middle here, when you look in the back, you are going to find behind there, you are going to find the memory center, the hippocampus. So that area here, and on the side outside that you look and see, that area is specialized in integrating auditory information, and also visual information is also it is connected to that center as well. So the medial temporal cortex temporarily stores these representations of the information that has become now kind of representations, like images. And these images or representations take the form of firing patterns. So everything that you see or hear that is coming to your cortex through nerve impulses, firing patterns, and these firing patterns become remembered, become stored. And when you make the neurons fire in the same way, they will recreate the information. They will make you get back that memory. So that area, the medial temporal cortex, it transfers these representations to cortical areas, for example, that initially received them. Let me go back to the image here. So the idea is this is the medial temporal lobe, but deep in it, on the other side, they are going to find the hippocampus. On the outside here, you are going to find the auditory association cortex that works to integrate what you hear with what you already know with your previous knowledge. So this area, the hippocampus, as I mentioned, like the mail processing center or like the airport, everything converges on there, and this area, this hippocampus is connected with all storage sites in the cortex, and the information then goes back and forth between the storage site and between the hippocampus, and also there's a constant back and forth between the prefrontal cortex and the hippocampus as it continues to process the information. It is almost like the prefrontal cortex is like the supervisor that is supervising what is going on here to make sure that things are retained that are important for the person's functioning, and then they sort it out, and everything is organized and makes sense. So you can store things and know how to find them when you need them. Okay. So here again is the, basically the limbic system. It's kind of, the term limbic is falling out of favor, but we still have it there. It doesn't matter what you call the whole thing. But again, here's the amygdala, the hippocampus. Here is the thalamus, that is like the gating mechanism for sensory information as it comes in. It fine-tunes it. And then you have the basal ganglia and different structures are over here. So encoding, taking in of new information and analyzing that information and connecting it or sending it for transfer to the storage sites, that requires, number one, emotional engagement, for the process to be conducted and to be actually to happen. You have to have emotional engagement. You have to be emotionally invested, interested. And that emotional engagement is going to create the motivation. And the motivation is going to cause you to have goals. Why am I doing this? Or I would like to do this. Once you have the goal, you have the motivation. I mean, you are going to build the goal. How do I get there? How do I achieve what I am motivated to achieve? And then you deploy your executive functioning skills, which are a set of skills dedicated to making goals and to plan for the future and to, look for resources that enable you to achieve that goal and monitor your own performance as you move and progress toward the goal. So encoding requires emotional engagement and motivation. First thing, they have to be deployed very early. Encoding or taking in new information also requires the ability to make associations. For example, you have a new system, you receive like a set of numbers. That is your social security number. How do you make sure that it goes in? So you might associate this number with this date, or this number with this, something you already know. You associate information that is coming in with information that you already know. And that makes it easier for you to begin to actually evaluate the information and to take it in. And the more knowledge that you have, the more background knowledge you have, the faster and the more information that you can process. Encoding requires attention. It is required for perceptual and for post-perceptual encoding and retrieval. For example, once you figure out the concept, you are aware of that information, then you need that in order, that perception part. You need attention for it. And after you perceive the information, make sense of it, you also want to break it down, interpret it, make sense of it, elaborate on that information, do more thinking, more in-depth analysis of it. That is post-perceptual encoding. And then retrieval. You need also attention in order to retrieve information. Retrieval means it implies intention. You retrieve something on demand. So when the information has a personal touch to it, when the information is emotionally relevant to the individual, that is going to increase the motivation. And that is going to make the information encoded on a deeper level. And it will make the information, for example, have a higher level of importance. For example, you are driving on the highway and see how many cars pass by you, left and right. Now you have a certain kind of car, a certain model, a certain, color. And all of a sudden, among this sea of cars on your left and right, you are going to look and you find the car like your own. How interested are you? How motivated are you to observe this car? To kind of look at it and, from far, to imagine yourself driving the car and say, oh, this is how people see me when I'm driving my car, and this is how the car looks, and so on. So you are going to be more attentive to it because it is personally meaningful to you, and that car is going to get more attention from you than any other car that's around. So that makes, again, the encoding of that information, that experience, is going to have, higher importance. And then you can use this as an example. If you are teaching a lesson, for example, and you can make it meaningful to the child. You can make the child, for example, instead of saying, okay, take this topic and write me 5 paragraphs on it. How about telling the child, find a topic, that has to do with something that you enjoy, and write me, something, 5 paragraphs. You get the same result, but the child is going to be, or the student will be more motivated because it is something that he or she chooses and enjoys to speak about. So that makes the task definitely much more enjoyable, and it will make it much more that will make the person more motivated and will make the learning experience ultimately more meaningful. So positive or emotions typically, produce a greater kind of result. That information that is socially, for example, emotionally relevant and significant to the individual, it has a high level of importance because at the same time, the person is relaxed, is not feeling like, stressed out more than usual. And that is going to enhance, to create an environment that is optimal for learning. When information is negative, it has a negative tone to it. It is going to narrow down the focus of your attention. It is going to cause you to not to pay attention to the details. Because negative emotions make the person kind of feel threatened, and that is going to narrow the focus of their attention, and they would have difficulty focusing on the details that are going around. I'm going to touch upon neurotransmitters and neuromodulators. These are the chemicals that facilitate the transmission of information from one neuron to the other, along the synapse. So I'll just touch upon the major ones, especially the ones that assist in the process of encoding, because every phase requires its own neurotransmitters. So these include the stress hormones. Or you say stress. I just mentioned, stress, higher levels of stress aren't good. But you have to have some kind of stress in order to learn new stuff. You have to feel challenged to some extent. If the information is all completely, something you already know, so what is the purpose? But if you go very, very high, you make the information more complex and beyond the person's ability, you challenge them too much, that also defeats the purpose. It will make the person lose motivation, because that information now, the complexity level is greater. That means you are going to put more cognitive resources. And if you give us the choice, we are going to do everything to avoid having the brain work very hard. That is mentally taxing. That is physically exhausting. So you want to identify the best level where the individual is able to perform really well, and then go above it a little bit. Challenge them, but not too much. So, for example, if you challenge them like 15% or 20%, that is reasonable. But if you challenge them 50%, give them something that is 50% harder, than they can manage, that is going to be really, really intense, and that could backfire. They would not be able to relate to that information as much, and you will lose their motivation. So find the ultimate best level for the individual where you can challenge them, not like to some extent, and not to be challenging them too much, just the right. When Goldilocks says it was just, I mean, the bears just right. And then you go from there. So you need these stress hormones to enable you to be up and alert and kind of awake, and you have the attention and the skills that are needed. Dopamine, norepinephrine, acetylcholine, cortisol. So these neurotransmitters work together in order to kind of facilitate the encoding process. Dopamine is the chemical or the neurotransmitter that enables us to feel happy. And when someone, for example, imagine a student working on a math problem, and she is kind of, well, let's say he is struggling, with the problem and trying to answer it, and trying and solving and not coming, with the right solution. And so then he tries again and gets the first step right and the second step right, and then then he's gonna add another step, and then he looks and he comes up with the solution. That feeling that he is going to feel when he identifies the solution, that satisfaction, inner satisfaction, that is the result of dopamine. It is what makes us feel this way. The same socially. You hold the bag in your right hand, and you hold a bunch of books in your left hand. You can't open the door. And you try to figure out how you are going to open the door, turn around, push it with your back, whatever. And then at the right moment, someone opens the door. A student just opens the door for you, a classroom door, and then you just kind of, thank you so much. And you saved the day. And that person is going to get a rush of dopamine, is going to feel the pleasure, the happiness, because he or she did something that kind of was good and made someone praise them. So that is going to give you that joy. Now, the good thing is, when we experience release of dopamine in these situations, the circuits that metabolize dopamine and that use dopamine, they have memory. They remember the pattern of release, and they associate that with the event that triggered the release, so that when you kind of, it doesn't take much to induce the same system. When you go and think about the event, just remember it here, it is going to induce more dopamine to be released. When you see the situation again, so every time that system works, that is going to kind of make the memory of the release of the dopamine stay longer, and that becomes a pattern, and then you will learn how to anticipate things and to be ready to release dopamine at the right time. There's some research to suggest that people with attention deficit hyperactivity disorder, they have difficulty, especially hyperactive, difficulty releasing that dopamine at the right time. So something happens, and then they don't, they kind of don't care much, because the event happens and the time passes, and then dopamine is released after. So it is not associated with what is happening at the moment. So that delay in releasing the dopamine in connection with the action, it is creating that discrepancy that is creating lack of motivation, and the person with attention deficit or hyperactivity might be kind of, not having that motivation. And they don't have the motivation, they wouldn't have the ability to direct their goals, to create goals, to have executive functioning skills, to regulate, when to do something, how to achieve their goal, how to organize their time. And it continues, like one step leading to the other. Norepinephrine and is also important in terms of elevating your kind of physical kind of energy level, and enabling you to have that optimal kind of level of not very stressed, but able to kind of have the arousal, what we call arousal in neurology, like of, in other words, the opposite of sleeping, opposite of hypoactivity, and so on. Acetylcholine is important for movement, is important for basically maintaining the functioning of the neuron itself. Cortisol is gonna elevate your blood pressure a little bit, gives you that energy boost to continue to send blood to the brain, and to kind of feel in that alertness, that sense of arousal, that you are up and alert and focused. So when you look at learning in a context, for example, certain contexts where you bond with others, for example, you have how does learning goes, and how do you learn from the social experiences with others, and how you connect with others. Learning in the presence of other people is more meaningful than learning by yourself. Even physical exercise, you can do the same set of exercises by yourself, and you can do them the same with a group, and the effect with a group is more significant, because the social effect is going to create the motivation, and it's going to create for example, and physical activity, the distraction from your physical activity, and also it's going to create the persistence, and you want to do it because you are encouraged by others, and you keep continuing doing it more, and you have the ability to continue it for a longer time, in addition to having that sense of you are not under stress, you are relaxed when you are in a positive social environment. Serotonin is essential for learning. It is essential for social bonding. Serotonin is a neurotransmitter that makes you kind of relaxed. It makes you basically like you need it when you are ready to sleep. It is what makes you dream. So it is actually, it enables and it helps dopamine to do its work. Oxytocin is a social bonding hormone, and serves as almost like a neurotransmitter or neuromodulator. So, for example, when you are you have a child and you touch the child, and you just for example hug the child, that is going to make the child release more oxytocin. So experiments show that children who are kind of have a good proximity with their parents, and they hug them and kiss them and kind of touch them, they grow physically, they grow well. And children who don't have that as much, their growth is stunted, it is not as robust. Children, when they have positive social experiences, they release oxytocin, and that facilitates memory for social events and memory for positive emotions and so on. So that is going to then create the longing to be with the group, the association with the group belonging. The only thing is that research shows when that oxytocin, it improves the person's ability to be with a group and to be adjusted and to feel happy and so on within the group dynamics. However, in some cases, it can cause the person to be aggressive toward someone who's coming from outside. So you are in a group, for example, let's say all of you, everyone is white, and now, everyone is bonding together and so on. And then all of a sudden, someone different comes into like your neighborhood or your group, and then people are just, oh, that's an invader. This is an offending party. This is that, and that stranger kind of component that is going to could lead to aggression, and do different kinds of different levels to kind of deal with that so-called invader. This is animal behavior, and humans, of course, have the ability they have knowledge when they know more and know how their brains work. They avoid these basic animal instincts, and this is why we are different than animals. Dopamine, again, is important for movement. It is important basically for emotional regulation. Deficiencies in it is gonna cause is gonna cause depression, and so it's important. Dopamine is important for movement, for mood regulation, for learning, for motivation. So all of those neurotransmitters are actively released in encoding, during encoding. Dopamine, DA, assigns motivational value, salience to a reward. Like, you are working toward a degree, and, kind of when you think about your goal, oh, I'm going to do this paper, I'm going to get this course, I'm going to get this degree. Every time you think about it, it increases your motivation. And it makes what you are working toward, it makes it more meaningful. So what do we take? How do we take this research finding, and translate it into something in the classroom? This is a skill that I hope that you can learn as you go. We remind students of their goals. When you become a doctor, you are going to be working with, with this and this and that. So when you look at the brain, this is what it does, and you study, and the person would have a goal, and that is going to make that information meaningful, and it will increase their motivation. You keep reminding, I am working on, say, for example, a 5-paragraph essay, and my goal is, maybe it could be, to really do a good job, or to finish a rough draft, or to get an A, whatever my goal is. We need to refresh, we need to remind the student of the goal he or she is working toward. And as you keep doing that, that is going to make it more relevant, that goal, and they can imagine themselves achieving that goal, and that continues to feed their motivation. Dopamine increases the drive toward the reward, the reward that you get when you achieve the goal. So you keep the goal in focus, and as you keep seeing yourself in your imagination at the end with the goal achieved, that is your reward, whatever that reward is. So oxytocin and dopamine act on the pathway together, to shape social motivation and to influence expression of affiliative behavior. Affiliative meaning to be with the group, to have group belonging. So that is achieved through oxytocin and dopamine together. The forming of new memories, establishing, taking experiences and making them, putting these experiences in a form that you can recall after. It requires the building of new networks, and it requires modulation of synaptic connectivity. You have to kind of regulate the connectivity between one part and another, one circuit and another, one neuron and another. In order to achieve that, you have to have neurotransmitters, and you have to have the prefrontal cortex engaged. This is the prefrontal cortex. So a piece of that line is covered by this image. You can go to your slides and just minimize it, and you'll be able to access it. So the prefrontal cortex, that is a critical area that, again, it mediates or controls your motivation and your decision making, your judgment, your attention, working memory. It is really, really the powerhouse of your entire brain and your entire voluntary control over things, or voluntary action as well. So it regulates your behavior, it regulates your learning, your memory, and it depends on 4 primary neurotransmitters, the prefrontal cortex. It is dopamine, norepinephrine, serotonin, and acetylcholine. So acetylcholine it has pathways. The adjective from acetylcholine is cholinergic, so something that has to do with the acetylcholine. So cholinergic neurons that use acetylcholine in the basal forebrain. Let's see the basal forebrain right here, this area here. So the brain, just as it goes like this, it just kind of bends over. So that's the basal forebrain. So the dopamine, the cholinergic system there projects. It means it goes from that point down to another location, and it sends commands, it sends directions to the hippocampus and to the thalamus, to the olfactory bulb, which is the smell, the nerve for smell, and the nucleus basilis minor. So basically, this network is quite vast, and you can trace cholinergic system here and in the orange, and you can see now, all of this is part of the this is the basal forebrain. So it just goes, the pathway goes all over the frontal cortex and the parietal cortex and to the visual cortex. All of this is part of the acetylcholine pathways circuit. And then it goes again into the limbic system, again here. So it's quite significant. And you can see now dopamine pathways, the red ones, they are in the an area called the ventral tegmentum. You don't have to know the specific details really too much, but just generally. And then these project to the prefrontal cortex, and they project also to the basilis, to the basal forebrain here, and they project also to the amygdala. So you can see now where these pathways send signals there, and you can see the basal forebrain sending signals all over basically. So these neurons that use acetylcholine are also the primary source of the cholinergic innervation of the cortex and the amygdala. Cortex is where information is stored. The amygdala is your emotional processing system, especially fear. The cholinergic neurons in the basal forebrain, they project mostly, like I mentioned, to the hippocampus, where the memory processing center is, the thalamus. That is your gating mechanism. For example, if you are hearing something and it comes to that level and it is not loud enough, you are in a noisy situation, the thalamus is going to enhance and amplify that signal even up to 100 times, so that you can kind of make sense of it. And then, so these structures are part of the cholinergic system, so that you can imagine that smell is part of that, and you can imagine that, for example, where is it? Motivation, and also emotional regulation. So the prefrontal cortex contains dopamine. It gets its dopamine from the area that's called the ventral tegmental area. So the ventral tegmental area, let me show you here, and that's part of the brain stem, but higher. And it produces this dopamine, and then it sends it to, and that is basically, so sometimes called the ventral tegmentum also. It has multiple receptors in the prefrontal cortex, because you need it here again. You regulate your emotions also here. You regulate your working memory, your attention, and that dopamine is critical to that, and your movement as well. The prefrontal cortex also directs. So see, this is where it receives it receives the dopamine, but it directs its pathways that use dopamine are directed to an area called the periaqueductal gray, that is basically in the core of the brain, and that is going to help us to control our attention and direct our attention in the way that we want, and to switch attention from one task to another. Dopamine also, it doesn't contribute to inhibitory control of the, for example, of reversal learning. What does that mean? Reversal learning, say for example, you go and follow a route going from here to Framingham, from your location to Framingham. Now, as you want to kind of think, how do I reverse back my road? Come back. How do I reverse my route? And you can kind of trace the steps. In that case, dopamine is not involved in reversal learning. So again, this is the part of the dopamine pathways here, and the ventral tegmental area would be around here, and part of the dopamine is used in the basal ganglia that mediate your movement, and people who have difficulty, who have depleted, or who don't have enough dopamine, or are not circulating it right, they are going to have Parkinson's disease. People who have excessive dopamine, they are going to have schizophrenia. People who have Parkinson's disease and they take L-Dopa, like dopamine medications. And when these dopamine medications exceed a certain level, the required level, they could actually induce symptoms of schizophrenia. So here's the dopaminergic system, and you could see that it goes to the prefrontal cortex, it goes to the parts of the somatosensory system, and again it goes also inside of the limbic system. So it is, you see now how it contributes to learning, to memory, to motivation, to movement, to emotional regulation. Norepinephrine is produced by an area called the locus coeruleus, and that is the primary site for the projection of norepinephrine to the prefrontal cortex. So the prefrontal cortex doesn't make its own norepinephrine or dopamine, it gets them from somewhere else. The projections, from the medial prefrontal cortex goes down to the locus coeruleus, which is in the brain stem, basically. So the activity of norepinephrine, that's very important. The activity influences all self-regulation functions. So, for example, including set shifting, attention, inhibitory control, reversal learning. That is essential when you need norepinephrine for these skills. And this is a pathway. The locus coeruleus is in the brainstem, and you can see now how the pathways continue and go to the basically the entire, just about the entire cortex, even in the cerebellum. Serotonin is a neurotransmitter that is produced by in an area called the raphe that project to the cerebral cortex and the hippocampus, an area called septum, and an area, and the amygdala, of course. So here's the system here, dopamine pathways, and you can see all the areas that are named are here. So we want to know, very important, we want to know what is the function exactly of serotonin. So, and also, as I mentioned, it interacts with dopamine, but it also interacts with acetylcholine. And serotonin plays a significant role in cognitive functions, including learning and memory. It is critical for activating your inhibitory control, your inhibition, and also reversal learning. This reversal learning is very important for analyzing information, for processing information, new information. So this is the pathway again, and you go back again and look at the limbic system. It's in the heart of everything. So you have memory, mood, processing of sleep, memory processing, sleep, cognition, serotonin. Serotonin, before I close, you can try this for yourself. It is found in poultry, in red meat, and found in milk products. Like if you have like cereal with milk before you sleep, or if you have say yogurt or some milk product at night, you are likely to have a dream, because the milk or milk products have tryptophan, and tryptophan has it breaks down into serotonin, and serotonin, is basically a neurotransmitter. If you have chicken, if you have beef, you are more likely to dream at night, especially for dinner, because of the tryptophan that breaks down into serotonin. So when we speak about consolidation, we are going to learn how consolidation occurs while you are sleeping. Okay. Thank you. These are some references, some selected references, and thank you. Next, we'll have section 3.